And finally, we're going to take a look at what they call the evaluation section, which would include your conclusion and evaluation here. And again, with the actual criteria in the IA, they're not using this point system. Basically, I think the evaluation in the end is worth, let's see, 6 points total. So that doesn't line up with my 3, 2, 1 numbers over here. But what I've done is I've split up this level into 4 different bits. So this is written up as the top level. If you score everything up here, then you're capable of getting, let's say, 5 to 6 points is what they're saying. But if you're aiming to satisfy each one of these separately, and then you can maximize your chance of getting the highest possible score. So we try to break these down and added some extra little bullet points here. So this first column here in the evaluation section is about your conclusion. And your conclusion in an experiment can include some of the analysis of your data. So you can sometimes include that with your conclusions. Some people like to do the data analysis first and then draw the conclusions and then kind of go back to the data analysis. So the data analysis means the statistical tests that you did or calculating the averages. Remember in the last video we said averages are probably not enough. That's just basic processing. You should be able to go further with that, with the graph and actually analyzing correlation coefficient or something like that, drawing a line of best fit if it's appropriate for your particular investigation. Detailed conclusion, described, justified, which is entirely relevant to the research question, fully supported by the data. In a previous video for the old syllabus, I made up this thing called BRVSH, which sounded like brush, but with a cool V in the center. There's a lot of bands doing that these days. So where B stands for biological explanations are in there, R stands for research is included, V stands for values are discussed, S stands for statistical tests are discussed as well too, and then H means reference back to the hypothesis. That depends on if you started with writing a hypothesis or not. So if not, then well, you don't get this cool brush thing. Using appropriate citation methods, whatever you're using, MLA, APA, just be consistent throughout. You should have an extensive reference section at the end with properly cited references that you're actually using that come from reasonable and reliable sources and not like Joe's favorite science goodies website and stuff about the universe or something like that. Okay, make sure it's a decent scientific literature that you're actually using. Okay, anything else to say here? Let's see. Yeah, obviously the stuff that you're talking about here has to actually follow on. Oh my gosh, has to actually follow on from your data and your analysis as well too. So if you're saying what's not actually shown in your graph, then that's not going to be good, right? Can't make stuff up. So for the evaluation section, oh yeah, so linking the citations, that kind of leads into the second one. So you can break it down a little bit. A conclusion is correctly described and justified through relevant comparison to this accepted scientific context. So this, I kind of just copy and paste the same thing. I said there's a little bit of redundancy going on because these things kind of go together, but make sure you are doing some research and comparing what people have found before with what you've actually found here. It's highly unlikely that you're doing something which no one else has tried before. If it is, you got to check to make sure that your question isn't completely bogus or you're testing something that really has no reason that it needs to be tested. Because if you've done your homework, then there's no way that you would expect that, the number of fingers on my left hand held up determine the rate of photosynthesis for a plant. It's just not a worthy question to ask. So there should be stuff out there, so make sure you're actually referencing it properly and not just paraphrasing and using it as your own words. That's plagiarism. People actually plagiarize without realizing they're plagiarizing. So be really careful about that and make sure to put, where this stuff came from. Marzano, whatever, and the year that it was published, 2004, if it supports what you're saying. So blah, 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 Marzano 2004. According to Johns 2012, blah, 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 blah. You can talk about that. Okay, these last two columns are about the evaluation. This is the toughest thing to do, so I would pay close attention to all these little boxes down here, please. Strengths and weaknesses of the investigation, such as limitations of the data and sources of error, are discussed and provide evidence of a clear understanding of the methodological issues. Did I say that right? Methodological issues involved in establishing the conclusion. In other words, you are criticizing your procedure and your plan and your research question approach. You are criticizing the controlled variables or the lack of controlling those variables. You are not talking about why you failed at doing something because you're not good at following instructions. Assuming that you followed instructions and someone else competent out there is following instructions, what can you criticize about your method? Problems with the plan is what you should be thinking about. Problems with the plan. And those are the changes that you should be talking about. I would put it in a table where one side it says limitations to the experiment and the other side says suggestions. And that will address this side. This side is about realistic suggestions and this side is about the limitations you identified. This table should be extensive and you should write with paragraphs explaining what that limitation is. If there's a controlled variable that you should have considered that you didn't actually consider, then write down what that is. Put down what the suggestion is and make sure that's detailed as well too. So more focus on the limitations of the experimental design. Sources of error. In the previous video, we talked about random versus systematic errors. Make sure you know the difference between random and systematic errors and actually include that in your particular lab report. Are they things that you could have avoided through better control of variables, or are they things that you could have avoided through choosing your instruments more wisely? Here it says avoid talking about human errors. Let's see. Are there more weaknesses that need to be discussed? Additional trials. That's a great freebie you can always talk about. The more trials you do, the more precise your readings could be, and you can eliminate outliers and you can address the issue of precision versus accuracy. Your suggestions should be realistic, not just try harder. If you say find a better tool, don't say a better stopwatch or a longer thermometer. I don't know what that means, but make sure you're suggesting what a real tool is. Don't make things up. Like, look it up, find out the names of the actual instruments that are out there that could actually help you measure light intensity instead of what you did in the experiment, which was just to say it's kind of dark or it's kind of bright. So how can you actually quantify those things and help reveal some more information to make your data more reliable and therefore help you answer your research question a little bit better? So yet again, this evaluation part, one of the toughest things to do. Make sure you spend a lot of time doing that properly. Okay, good job. Gotta go. Thanks very much.